The olive ridley sea turtle is listed under the Endangered Species Act as a threatened species. This species of sea turtle nests in the Pacific, Atlantic, and Indian Oceans, and exhibits a mass nesting behavior at only few beaches within its nesting range. My field site is in Ostional on the northern Pacific coast of Costa Rica. Here, mass nesting events, or arrabadas, have been estimated at up to 500,000 nesting females over a period of only 7 days. This behavior is believed to be a predator satiation technique in which the abundance of eggs overwhelms the large population of predators. Despite the large number of nesting females, concern remains that the low hatching success at this beach is not enough to sustain the population long term. Several studies have suggested that embryo mortality is associated with the high microbial load resulting from the decomposition of eggs broken by subsequent nesting turtles. Thus, a legalized community-based egg harvest program is aimed at reducing the number of nests destroyed while providing the funds to support local infrastructure and family income. This harvest collects eggs within the first several days of each mass nesting event, removing nests that would ultimately be dug up by nesting females within the following days. However, no previous research has directly quantified microbial abundance and the associated direct and/or indirect effects on hatching success in situ. My study aims to determine the impact of microbial abundance on hatching success by applying experimental treatments to reduce the microbial abundance of the sand into which nests are relocated. Microbes, such as bacteria and fungi, compete for the same resources that sea turtle embryos require for development. Microbes require oxygen to consume organic matter and release heat in the process of metabolizing. Sea turtle embryos also consume oxygen to metabolize and develop under specific temperature requirements. This competition for resources leads me to the hypothesis that, as microbial abundance in the nest sand increases, hatching success decreases due to the limitations in resources. My study utilizes a hatchery-like setup of plots made up of sand treated with various antimicrobials to provide an in-situ nest environment under different microbial conditions. In one hatchery, sand is removed and treated with a bleach solution for 24 hours to decrease the microbial abundance of the sand before it is rinsed and replaced into plots where nests are relocated. In the other hatchery, sand is treated with a topical application of seawater with the intent of altering the osmotic environment beyond the tolerance limits of the microbial community. Nests are then relocated into these prepared plots within a single night of a mass nesting event. Samples of sand from the nest are taken for organic matter and molecular analysis, which will quantify microbial abundance with quantitative polymerase change reactions. Temperature data loggers and oxygen sampling also monitor relevant nest conditions throughout the incubation period, and nest excavations provide a quantification of embryonic development and hatching success. To better explain how the microbial abundance of nest sand might have an impact on sea turtles, let's look at the cross-section of a sea turtle nest in the ground. Under typical conditions, there is a relatively low amount of organic matter present for microbes to consume, and the competition between microbes and sea turtles is relatively equal. The diffusion of oxygen provides a net inward movement of oxygen to the nest, and the metabolizing embryos within the nest release heat outwards. However, when there is a large amount of organic matter, the microbes may be outcompeting the turtles. The microbial community becomes more active and consumes more oxygen, disrupting the oxygen gradient and depleting the nest of oxygen. Additionally, the microbial metabolism releases heat that increases nest temperatures. In fact, microbial abundance alone is likely not as important in disrupting the nest environment as is the microbial activity. Preliminary results thus far confirm the hypothesis that microbial activity is having a negative effect on hatching success. Briefly, a pilot study on the effect of various sand treatments on hatching success revealed a marked increase in hatching success for nests relocated into sand treated with a bleach solution, and a significant decrease in hatching success in areas suspected to have the highest abundance of microbes. Second, nests located in the area with the lowest hatching success also have significantly more organic matter content in comparison to other nest locations. Nests that failed also had significantly higher nest temperatures and were exposed to temperatures above the lethal limit of 35 degrees Celsius for a significantly longer duration of time. More importantly, nests with the lowest hatching success also had significantly lower oxygen content, especially early in the incubation period. Because embryonic development is limited within the first half of the incubation period, typical sea turtle nests do not exhibit a drop in oxygen until later in the incubation period. This drastic drop in oxygen therefore suggests the microbial community within the sand is responsible for this decrease in oxygen. Furthermore, microscopy counts of natural nest sand have confirmed a significant negative correlation between the abundance of bacteria per gram of nest sand and hatching success. My research is now focusing on simplifying sand treatments, targeting sand with higher microbial abundance, increasing sample size, and including controls to measure the microbial activity within beach sand outside of sea turtle nests. Should experimental treatments prove successful, this could be applied as a management technique to improve hatching success. In fact, the topical application of seawater is intended as a trial run for its use as a natural antimicrobial that can be applied to the beach as a community conservation activity at Ostional. My field site also serves as a model system of the potential impact of erosion and anthropogenic organic loading on sea turtle nesting success. These treatments would be especially useful for conservation programs that are experiencing microbial infestations in the hatcheries that they depend on due to erosion. Climate change is also an issue of concern because microbial decomposition is already decreasing nest oxygen content and increasing temperatures, thus having a negative impact on hatching success. Further increases in temperature due to climate change could accelerate microbial decomposition rates and create a positive feedback loop leading to more drastic decreases in hatching success. Ultimately, this research will help ensure the sustainability of the egg harvest at Ostional, the future of this natural phenomenon, and hatching success at nesting beaches around the world. I could probably have done a presentation solely for the purpose of thanking my funding sources and support, but for the sake of time, I just want to express strongly that this research would not be possible without the funding, logistical, and moral support that I have received. Various organizations here at the College of Charleston provided me with the small grants necessary to kickstart this research. The support of the National Geographic Young Explorers Grant and a portion of a grant from the Marine Turtle Conservation Act supported my pilot study last year. This year, my research has been supported by the National Science Foundation's Graduate Research Fellowship and a grant from the Patty Foundation, as well as several private donations made through a crowdfunding fundraiser at GoFundMe.com. I also received a lot of on-site logistical support from many organizations in Costa Rica. And I can't possibly leave out all of the individuals that have directly influenced my research interests, experimental design, and those who have assisted in carrying it out. My committee, my advisor Craig Plant, my mentor and the inspiration behind this project, Roldan Valverde, everyone in the Plant Lab, all of my research assistants here in Charleston and in Costa Rica, the sea turtle community for advice and logistical support, and countless numbers of volunteers from around the world who have been troopers in putting up with the large amounts of manual labor required of them. In the end, it's all worth it for the turtles.